So today we are going to see the fertilization chapter. This is the last chapter. So as you know, the fertilization is the fusion of the sperm cell and the egg or the oocyte to give the zygote. Okay, the zygote, which is a diploid cell and will be a totipotent stem cell. So the zygote is the stem cell that can give all the other type of cells in the body, and that's why it is called a totipotent. So it has a total potential of generation for the other cells. That's why it is toti from total and potent from potential. So it's a totipotent stem cell, the zygote. Now the fusion after the during the fertilization, we will have the fusion of the male and the female gametes, and due to this fusion will restore the diploid status of the species. Since the female gametes are haploids and the male gametes are haploids, when they fuse, we will have a diploid cell. Okay, and that's how we will be conserving the diploid status of the species. And this fusion will create new assortment of genes inherited by the maternal and paternal chromosomes. So because this cell, the zygote, will have chromosomes from the mother and from the father, so it will have a unique assortment of genes. Okay, and it will be a specific and unique genetic heritage for each individual. Now, as you know, before that we have the fertilization, we had the ovulation, and after the ovulation, the ovulated oocyte should be transported to the site of fertilization. And as we have discussed previously, the site of fertilization is the ampulla in the fallopian tube. So it will be here in this region, in the ampulla of the fallopian tube, the oviduct ampulla. So this will be the site of fertilization. So in order to have the fertilization here, we should have the transport of the oocyte to the ampulla, from the ovary to the ampulla, and also the transport of the sperm cells from the vagina that were ejaculated in the vagina. So the sperm cell will be transported from the vagina to the ampulla. Now, regarding the oocyte transport, also, as you know, that the oocyte was ovulated as an oocyte 2, يعني an oocyte blocked in the metaphase 2 of the meiosis. It is expulsed in this form from the ovary, and when it is expulsed, it is not all, as you know, not all the follicle is expulsed, but only the oocyte. This oocyte 2 will be expulsed from the ovary. But with the surrounding granulosa cells, which are the corona radiata and the cumulus oophorus. So when we will have the ovulation, the oocyte will be expulsed with the cumulus oophorus cells, granulosa cells, and the corona radiata cells, and with the follicular fluid. Okay, but the remaining part of the follicle will remain in the ovary and will not be expulsed with the oocyte. And then this oocyte too, when it is expulsed, it will be captured by the fimbriae of the fallopian tube, and due to the movements of those fimbriae and to the we'll have a contraction of the fimbriae to capture this oocyte, and also we will have as you know, we have here inside of this fallopian tube we have epithelial cells, ciliated epithelial cells, and those cilia, the beating of those cilia will be stimulated by the estrogen, the high estrogen levels, the pre-ovulatory high estrogen levels. So the beatings of those cilia will help also in transporting this oocyte, this expulsed oocyte, to the site of fertilization. So from one side, we have also muscles here, okay, surrounding the fallopian tube. So we will have contraction of those muscles and to the beating of the cilia. So due to those both events, the oocyte can be transported from the ovary and from the site of the ovulation to the ampulla, the site of the fertilization. Okay. So the peristaltic movement, they are the contraction and relaxing movement of the muscles of the fallopian tube. We will have contractions of the muscles here that will help in the movement in pushing the oocyte to the ampulla, and also the beats of the cilia of the epithelial cells inside of the fallopian tube that will help the oocyte to reach the ampulla. Now also the sperm, as we have said, should be transported from the site where it has been deposited, of ejaculation, and from the vagina to the ampulla. Now regarding the sperm transport, during the ejaculation, the male ejaculates 50 to 600 million sperm per ejaculation. Okay, so the male will be ejaculating those sperm cells in the seminal fluid, and as we said, we call the sperm cells plus the seminal fluid, we call it the semen. So the male will be ejaculating a few millimeters of semen that will contain 50 to 600 million sperm cells in the vagina of the female, and only 1% of those sperm cells will cross the uterine cavity to reach the fallopian tube. So only few of them will reach the fallopian tubes to fertilize the oocyte. Okay. Now, the first step when the sperm cells are ejaculated in the female genital tract, they are ejaculated or deposited in the vagina. So first of all, they will be in the vagina, and just after the insemination, and just after their ejaculation in the vagina, the seminal plasma, the fluid surrounding those sperm cells, is called the seminal plasma. So this seminal plasma will be coagulated, and this coagulation is due to proteins in this seminal plasma. The sperm cells, they are in the seminal plasma, and this seminal plasma contains proteins that will allow this coagulation. So just after their insemination in the vagina, this seminal plasma will be coagulated, and 30 minutes later, this ejaculate will be liquefied again. So after their coagulation, they will be liquefied. This seminal plasma will be liquefied, and also this liquefaction is made possible due to the presence of other enzymes in the seminal plasma itself, and that will help in the liquefying the seminal plasma. So after clotting, the clotting of the seminal plasma, 30 minutes later, we will have this seminal plasma that will be liquefied, and this will allow the release of the fully motile spermatozoa. Now, just why it is coagulated and then liquefied, they don't know exactly why, the reason why, but it seems it is important for the good process of fertilization. So then it will be liquefied, and this liquefaction will allow the release of the fully motile spermatozoa. Now, the spermatozoa, they are still in the vagina. They will move to the cervix, and as you know, in the pre-ovulatory period or the peri-ovulatory period, around the ovulation the ovulation period. Okay, this cervical mucus is fluid. As we said, it is aqueous and abundant and fluid, not thick. So it is fluid, and this status or this state of the cervical mucus will help those sperm cells to move further into the cervix and to reach the uterus. Okay, and so this state of the cervical mucus will promote the penetration of a few million of those sperm cells in the cervical canal. Now they were 50 to 600 million. Now only few millions will reach the cervical canal. And the sperms that are, that could not cross this cervical mucus, or that were not selected by the mucus, they will be destroyed by the acidic pH of the vagina, and they will remain in the vagina. And the vagina has an acidic pH, and as you know, the sperm cells have alkaline pH, and they don't in fact, they cannot handle the acidic pH. So if they remain for a long period in an acidic pH, they will die. So if they cannot cross this vagina, if they cannot cross the cervical mucus to reach the uterus, they will die in the vagina due to the acidic pH of this vagina. So that's how the cervical mucus will be the first physiological barrier that will regulate the access of the spermatozoa to the uterine cavity. So now the cervical mucus is the first physiological barrier that will regulate the access of the spermatozoa to the uterine cavity, because if they can cross it, they will reach the uterine cavity. If they cannot cross it, they will die due to the acidic pH of the vagina. Okay, so the cervical mucus is the first physiological barrier for the sperm cells during the fertilization process. And if you remember, we said that this cervical mucus is fluid in the pre-ovulatory period, or around in the peri-ovulatory period, in the ovulatory period. And before and after the ovulation, it will be a mucus, and this thick mucus will not will constitute a true barrier for the passage of those sperm cells. Okay, but this thick mucus will help to protect the forming fetus, because it will constitute a barrier for the passage of the bacteria and microorganisms to the uterus. Okay, so it will be liquid and aqueous only in the peri-ovulatory period. Now, once they cross, yes.
Where does the liquefaction of the seminal fluid occur?
In the vagina.
Okay, doctor.
Now, once they cross the surface, once they cross the cervical mucus or the cervix, they should reach the uterus. Okay, and then from the uterus, they will go to the fallopian tube here, or the oviduct, what we call the oviduct. But now only a few thousands of those migrating sperm cells in the uterus will reach the fallopian tube. The others will die in the uterus, okay? Because in fact, during their migration in the uterus, a large number of sperm cells will remain captured in the uterine glands. Okay, they will be captured in the uterine glands. Then they can be released also to reach the fallopian tubes, but some of them will die because they will be phagocytosed by the polynuclear cells and the macrophage cells, by the white blood cells, okay, which will invade the uterine cavity 24 hours after insemination. So if they remain captured in those uterine glands and will not move from them. They will be phagocytosed by the polynuclear cells and the macrophages, and they will die. And that's how they will decrease in number, and only few thousand of those. If you remember, we said we had few millions that reached the uterus. From those few millions, sorry. From those few millions, we have few thousand that will succeed to pass this uterus and reach the fallopian tube. And once they reach the fallopian tube, they have another barrier here, which will be at the level of the uterotubal junction. Yani this junction between the uterus and the fallopian tube, this region here, secrete يعني produce some secretions that will constitute a barrier also for the passage of the sperm cells. Excuse me. So the secretions of the uterotubal junction will constitute a barrier for the passage of the sperm cells, and many of them will be stuck here and maintained here, and then they will die. And few thousand, only few thousands will succeed to pass this junction, this uterotubal junction, and will reach the ampulla. So finally, at the end of this migration of the sperm cells, only 100 to 300 sperm cells will can reach the ampulla and will gather around the oocyte, and all the others will die. Okay, so now in this way, this uterotubal junction is the second barrier regulating the number of spermatozoa reaching the oocyte. So the first barrier was the cervical mucus. The second barrier is the uterotubal junction with its secretions that will also can prevent the movement or the further progression of those sperm cells. So this will be the second barrier, the uterotubal junction. And at the end, only 100 to 300 spermatozoa can pass this barrier and will reach the ampulla and will gather around the oocyte.
دكتور بس مش لازم يكون الثيرد بارير؟ ما بنعتبر اليوترين جلاندز بارير؟
هن منهم barrier اليوترال glands لأنه هن they don't inhibit the migration of the sperm cells, but they will be phagocytosed in the uterus. يعني إذا بضلون كتير بالuterus they will be phagocytosed. Okay. So as a physical barrier يعني as هو mucus that is constituting a barrier. يعني the first barrier هي the mucus secreted by the cervix, and the second barrier is the mucus secreted by the uterotubal junction. هلا you are right إنه yes, a big number of them will die also in the uterus, but it's not a barrier. يعني منه حاجز من إنه يمرقوا sperm cells. Okay.
اوكي دكتور فهمت قصدك.
يعني هنا الميوكس لأنه it's inhibiting أو preventing يعني ما عم بيخليهم يمرقوا, we consider it as a barrier. هلا it's true in the uterus their number is decreasing also, but it is due to the phagocytosis, but we don't have a physical barrier. يعني we don't have something produced that inhibit their passage.
اوكي دكتور كلير.
Now, three factors are involved.
دكتور بس في حدا كان عم يحكي يمكن صوته واطي أو شيء.
Okay, sorry. Does anyone have a question?
لارا بتخيل.
Okay, I'm listening.
خلص دكتور مش الحال ميرسي.
Okay. And now the factors which are involved in the passage of the sperm cells to reach the oocytes are the motility of the sperm, the structure and composition of the cervical mucus, and the muscular activity of the vagina and the uterus. So those are the main factors involved in the passage, in the migration or in the transportation of the in the movement. And those are the main factors involved in the movement or the migration process of the sperm cells from the vagina to reach the oocyte. First of all, we have their motility, because if they are not motile enough, or they have a problem in their motility, they will not be able to reach the oocyte. Second, we have the structure and composition of the cervical mucus, because as we have said, during the periovulatory period, the cervical mucus is liquid and they can cross it, and but not if we are in the luteal phase of the ovarian cycle or in the early follicular phase, the cervical mucus will be thick and they will not be able to cross it. And the third element that will help in the movement and the passage of the sperm cells to reach the oocyte is the muscular activity of the vagina and the uterus, because we will have contractions of the muscles of the vagina and the uterus, and those contractions will be stimulated by the oxytocin produced by the female pituitary gland, and the prostaglandins present in the semen, that are present in the semen of the male. Okay, the sperm cells, they are in this seminal fluid or plasma. So once they are in this seminal plasma contain prostaglandins, and those prostaglandins will stimulate the contractions of the uterus and the uterine tubes, and those contractions will help in the movement of the sperm cells. Now we should have some modification. It's not enough. The migration of the sperm cells to the site of fertilization alone is not enough to have the fertilization. We should have some modification at the level of those sperm cells to allow this fertilization to occur. Okay, and those modifications that we should have on the sperm cells, they are called the capacitation. Okay, they will be referred to as the capacitation process. So this capacitation process will occur in the female genital tract, and they are transformations that will make the sperm cell able to recognize the zona pellucida of the oocyte. If we don't have those changes in the at the level of the sperm cell, the sperm cell will not be able to recognize the oocyte, the zona pellucida of the oocyte, and thus we will not have the fertilization. And if you remember, we said that during the maturation of the sperm cells in the male genital tract, they will be decapacitated. We said we had different changes that will occur to those sperm cells produced by the epididymis, by the testicles of the male. Okay, in the epididymis and in the seminal fluid, we will have many changes that will occur to them. And those changes, we call them the decapacitation, where we will have some proteins that will come and bind to the surface of the sperm cells, and some other glycoproteins. We had the cholesterol levels that will increase in the plasma membrane of the sperm cells, etc. And we said that all of those changes will prevent any reaction of the sperm cells with any type of cell. Okay, this was the decapacitation. Now, in the female genital tract, in fact, we want those sperm cells to interact with the oocyte. So we should, that's why it is the opposite of the decapacitation. And all the proteins that were, and the glycoproteins and the changes that have been added to those sperm cells in the male genital tract will be removed again in the female genital tract, and in order to help this sperm cell to interact, to recognize the zona pellucida of the female oocyte and to interact with this oocyte. So in order to have the capacitation, we should eliminate the seminal fluid. If we don't eliminate the seminal fluid, the capacitation will not occur. Okay, so first of all, we should have the elimination of the seminal fluid, and this elimination of the seminal fluid will occur in the cervix. Okay, the cervical mucus will so we will have the elimination of the seminal fluid, and then after the elimination of this fluid, only the sperm cells in the cervix and in the cervical mucus will enter the cervical canal. Okay, so in the cervical canal, to progress in the cervical canal, this seminal fluid should be removed, and it will be removed in the cervical mucus, and only the sperm cells alone, without their seminal fluid, will cross the cervical mucus and will reach the cervical canal. Okay, because those components of the seminal fluid will prevent the initiation of the acrosomal reaction and will prevent the capacitation also. Now, what are the elements involved in this capacitation? They are the secretions of the uterine and fallopian tubes. And the secretions of the uterus and the secretions of the fallopian tube are directly involved in the sperm survival and capacitation. And thus the capacitation will occur only in the uterus and the oviducts. The capacitation don't start in the cervix. We don't have a capacitation in the vagina. The capacitation takes place only in the uterus and in the fallopian tubes, and this is because of the secretions and the composition of the secretions of the uterus and the fallopian tubes that this capacitation is possible. Now, during the capacitation, we said that we will have changes in the plasma membrane of the sperm cell, and those changes include the removal of the protective proteins. As you remember, we had some protective proteins here, so those proteins should be removed. The removal of cholesterol, because also, if you remember, we had the cholesterol. Let me see. Okay, we said that during the maturation of the spermatozoa and the male genital tract, we had the addition of cholesterol in the lipid bilayer of the sperm cells. Okay, now in this capacitation process, we should remove those cholesterol molecules from this bilayer, and we should unmask the sperm receptors, because also during this decapacitation we had a protection of the sperm receptors here, and we should unmask those sperm receptors, because those sperm receptors will allow to the sperm cell to recognize the zona pellucida and to bind to it. So we have three major phenomenon that will occur during the capacitation. First of all, the removal of the protective proteins, from the surface of the sperm cell, from the sperm cell membrane or plasma membrane, the removal of the cholesterol molecules from this bilayer, from the cytoplasmic membrane يعني كمان, and the unmasking of the sperm receptors. First of all, we will talk about the removal of the protective proteins. So this is the sperm cell, the sperm cytoplasmic membrane. Okay, we have this bilayer here, with the phospholipid bilayer constituting the sperm cell cytoplasmic membrane. And during the decapacitation, we had the addition of those surface proteins that were added in the male genital tract. Now, during the capacitation, we should remove them. So, how will they be removed? Now, those proteins, as we said, they come from the epididymis and the seminal fluid. So those added proteins, they were added in the epididymis and the seminal fluid and should be removed. So this elimination will take place in the uterus and in the fallopian tubes. And in fact, due to the secretions of the uterus and the fallopian tubes, their secretions contains glycosaminoglycans and albumin. Okay, the glycosaminoglycans produced by the uterus and the fallopian tubes and the albumin also can bind to those surface proteins, and by binding to them, they will remove them from the surface of the sperm cells. Okay, and this action also was made possible due to the acidic pH of the uterus. So the acidic pH of the uterus will facilitate this detachment. Okay, so we have two important elements in the fallopian tube secretions and uterus secretions, which are the albumin and the glycosaminoglycans. They bind to those surface protein and remove them, and this is facilitated by the acidic pH of the uterus. The second step of the capacitation is the removal of the cholesterol. In fact, the cholesterol in the lipid bilayer, in this phospholipidic bilayer, will help to stabilize this membrane, will help in stabilizing the membrane of the sperm cells, and thus to inhibit any sort of interaction or any acrosomal reaction of this bilayer, of the sperm cell with any other cell. Now, during the capacitation, those cholesterol should be removed, and their removal is made possible to the presence of the albumin, also the albumin, and the high-density lipoprotein, okay, which are present in the uterotubular secretion, يعني in the uterus, and here the uterotubular secretion, يعني the secretions of the uterotubal here at the uterotubular junction. So the albumin is produced and the HDL are produced by the uterotubular secretions and will help to remove the cholesterol from the lipid bilayer, because the albumin and HDL will bind. So here you have the cholesterol fragments. So the albumin can bind to those cholesterol and remove them, and also the HDL, the high density lipoproteins. And as a consequence of the removal of this cholesterol from the lipid bilayer, we will have a decrease in the cholesterol levels, and thus the membrane will be more motile. يعني we will have, you know that the phospholipids in the membrane, they can have a certain motility, which provide mobility or a motility for this membrane. So by removing those cholesterol molecules, this will, as a consequence, we will have a mobility of the phospholipids in the phospholipid bilayer, and this will contribute to the membrane destabilization. So due to this motility of the phospholipids in the cytoplasmic membrane, we will have a membrane destabilization, and it will be less stable, and as a consequence of this destabilization, we will have the opening of ion channels on the cytoplasmic membrane. Because as you know, we have many types of ion channels on the cytoplasmic membrane. Here the ion channels, the sodium acid, the sodium H3O plus channels will open, the potassium bicarbonate channel, the channel of K plus slash HCO3 minus channels will open also. And due to this opening of those channels, we will have a change in the membrane a depolarization of the cytoplasmic membrane and an activation of the calcium channels. Okay, and this depolarization of the membrane and mostly the activation of the calcium channels and the entry of the calcium has very important consequences in the capacitation process. Okay.
دكتور
Yes.
بس الانترا ممبرين موبيليتي هي نفسها الفلويديتي تبع الممبرين.
Yes,
okay.
Fluidity, yes. So the removal of the cholesterol level will increase, if you want, the fluidity of the membrane and the fluidity of the phospholipids, and thus the cytoplasmic membrane. This will lead to its destabilization and the opening of the ion channels, the sodium, hydrogen, and potassium bicarbonate channels, and the activation of the calcium channels in consequence. So how the opening of the calcium channels will occur? We have first the activation of the ionic exchange, sodium H3O plus. Okay, when we put H plus, it's the acidic channel, the H3O plus. Normally in the cell, the sodium is mostly outside of the cell, and the potassium is inside. And when we will activate those channels, we have a channel for the sodium H3O plus. So when this channel is activated, the sodium that was at the outside of the cell will go inside, and in parallel, the H3O plus will go from the inside to the outside, and it will exit the cell. And in this way, the acidic ions will exit the cell, and this will lead to an increase of an intracellular pH. Since the acid ions went out of the cell, the pH inside of the cell will become more basic, will increase. And this alkalinization of the intracellular pH or of the cytoplasm will promote the opening of the calcium channel. So as a result of the alkalinization of the intracellular medium, we will have the opening of the calcium channels. This is from one side regarding the sodium acid channels. Now, the potassium bicarbonate channels also will activate the opening of the calcium channels. The potassium are mostly, as we said, inside of the cell, and the bicarbonate outside of the cell. So when we will have the opening of those channels, the potassium will go كان جوا رح يطلع لبرا, and those bicarbonate ions هن alkaline ions. Okay, they were outside, they will enter to the inside of the cell. Okay, and this will lead to an alkalinization of the cell, also of the intercellular medium, that will lead to the opening of the calcium channel, and also to a hyperpolarization of the sperm membrane. The sperm membrane will be more negatively charged from positively charged from the inside. Okay, and this will lead to the depolarization. Okay, this will lead to the hyperpolarization of the membrane. It will become negative. Okay, and due to this hyperpolarization, we will have also an opening of the calcium channels. So this calcium, this increased calcium, why we said that the sperm membrane will become more negative, لأنه the H3 the HCO3 minus ions entered inside of the cell, and the H plus ions went out of the cell. This will render the inside the cytoplasmic membrane of the sperm cell more negative, okay, and this will lead to the opening of the calcium channels also. So we will have the alkalinization of the cytoplasmic medium, of the intracellular medium, and also the hyperpolarization of the sperm membrane that will lead to the opening of the calcium channels. And this rise, we will have an opening of the calcium channels. So if you have an opening of the calcium channels, this means that the calcium can can enter inside of the cell, from the outside to the inside. So this will lead to an increase in the calcium levels inside of the sperm cell, and those increased calcium levels will induce the sperm motility. We will have a hyperactivated sperm, okay, it will be very active, more than it was normally. So those increased intracellular calcium levels in the sperm will induce the sperm motility, a hyperactivated sperm motility will facilitate this calcium, will facilitate the acrosomal reaction. We will see how now. And this calcium also will lead to the unmasking of the sperm receptors. So that's why this intracellular calcium, the rise in this intracellular calcium in the sperm cell is very important in the capacitation process. And this rise in the intracellular calcium was induced by the removal of the cholesterol. So this calcium and bicarbonate, the removal of the cholesterol induced the entry of calcium and bicarbonate ions into the sperm cell cytoplasm, and this will activate the adenylate cyclase, the enzyme, the adenylate cyclase. And this adenylate cyclase, in fact, look, the entry of the calcium and of the bicarbonate ion inside of the cytoplasm will activate this enzyme, the adenylate cyclase, which is also called adenylyl cyclase. Maybe in your books it is referred as adenylyl cyclase. So it is the same. The adenylate cyclase will be activated, and this enzyme transform the ATP into cyclic AMP. So the cyclic AMP will be thus produced as a consequence of the activation of this enzyme. So as a consequence of the entry of the calcium and bicarbonate, we will have the activation of the adenylate cyclase, which will this will induce the production of the cyclic AMP. And the cyclic AMP produce will activate some proteins inside of the cell, like the protein kinase A and the protein kinase C. Okay, I can't see it here. So we have the protein kinase C and A, and both of them will target other proteins involved in the motility of the sperm, so they will that's how the increase in the calcium level will hyperactivate the sperm motility, and they will induce facilitate the acrosomal reaction. They will activate other enzymes implicated in the acrosomal reaction. So this increase in calcium and bicarbonate activate the enzyme adenylate cyclase, which will induce the formation of the cyclic AMP from the ATP. And then we will have, as a consequence, this cyclic AMP will activate the protein kinase A. And this protein kinase A, from one side, it phosphorylates certain flagellar proteins, okay, that are associated with changes in motility. This protein kinase A will activate or will phosphorylate some proteins at the level of the flagella, and this phosphorylation of the flagellar proteins will induce the changes in the motility of the sperm cell, and that's how we will have an activation of the sperm motility. the sperm cell will become hyperactivated. It will have hyperactivated movement. And from another side, this protein kinase A will phosphorylate certain proteins like the heat shock proteins, okay, which are involved in weakening the cytoplasmic membrane of the sperm cell. So we will have an activation of certain enzymes or proteins that will weaken this cytoplasmic membrane of the sperm cell. And as a result of this weakening of the sperm cell, as you know, we have the acrosome that is directly present beneath. This is the acrosome, and this is the cytoplasmic membrane of the sperm cell. So the acrosome, due to the weakening of the cytoplasmic membrane, can fuse with the cytoplasmic membrane, and this will help in activating the acrosomal reaction. And as a consequence of the fusion of the acrosome with the cytoplasmic membrane, we will have pores that will be formed at the level of the cytoplasmic membrane and the acrosome, and thus we will have the release of the content of the acrosome, and the acrosomal reaction can happen. Okay. So due to the phosphorylation of certain proteins as the HSP, the heat shock proteins, we will have a weakening of the cytoplasmic membrane of the sperm cell, which will facilitate the acrosomal reaction, okay, after fixation on the zona pellucida. But the acrosomal reaction will only occur if we have a fixation on the zona pellucida. If we don't have a fixation on the zona pellucida, we cannot have an acrosomal reaction. But this calcium, this increase in the calcium and bicarbonate levels will weaken the cytoplasmic membrane, which will facilitate later the acrosomal reaction. Okay. And now we will talk about the unmasking of the sperm receptor during the capacitation of the sperm cell. So the unmasking of the sperm receptors is due to the redistribution of the phospholipids. So now we said that this fluidity, the removal of the cholesterol, will lead to some movement of the phospholipids at the level of the cytoplasmic membrane, and we will have a redistribution of those phospholipids. This redistribution of the phospholipids will lead to the unmasking of the sperm receptors, and some sperm receptors were masked, were covered, were not exposed to the outside, so they can recognize their target. Now, after this redistribution of the phospholipids, they will be unmasked or more exposed to the outside. Okay. And this unmasking of the sperm receptors is also due to the entry of the calcium, because this entry of the calcium modifies the oligosaccharide chain of the surface protein, and this modification of the oligosaccharide chains of the surface proteins will lead to the changes in the structure of proteins, and will make the receptors more accessible. Okay, so as a consequence of this entry of calcium, we will have modification of those oligosaccharide chains, the oligosaccharide chains of the surface proteins. Okay, and as a consequence of this removal of the oligosaccharide chain, the receptors can be unmasked, will be unmasked. And since the receptors are now accessible and unmasked, they can recognize and bind to the zona pellucida. ZP3 is a glycoprotein on the zona pellucida, but it is not the only glycoprotein that will be recognized by the sperm cell. We will talk about them later. Okay, so due to the unmasking of the receptors, they will be accessible and they can recognize and bind some zona pellucida glycoproteins, and the binding of the zona pellucida will induce the acrosomal reaction, يعني the release of the enzymes inside of the acrosome. Now the sperm binding to the zona pellucida is specific to the species, which means that the receptors present on the sperm cell, okay, are specific to the zona pellucida glycoproteins for the same species. And a sperm cell cannot recognize the zona pellucida of, cannot bind and recognize the zona pellucida of an oocyte from another species, because the proteins are different and we will not have a recognition between the receptors and the proteins or glycoproteins of the zona pellucida. So the sperm cells of a certain species will bind to the zona pellucida of the same species. Okay, they are specific for this species, and that's why we cannot have a fertilization between two different species of animals, for example. Okay, but the binding of the sperm cell to the egg is not specific to the species, but it is mostly the binding to the zona pellucida. Because during the fertilization, we will have the first step, which is the binding to the zona pellucida, and the second step, the binding to the oocyte itself. Okay, so it is the binding to the zona pellucida that is specific to the species. Now here we have, after the cholesterol removal by binding to the albumin and the HDL lipoproteins, we will have the opening of the potassium bicarbonate channel. The potassium will go out and the bicarbonate will enter, and this will alkalinize the medium and will hyperpolarize the cytoplasmic membrane, which will lead to the entry of the calcium and an increase of the intracellular calcium level in the sperm cell. So the entry of the calcium and the bicarbonate and also of the bicarbonate will activate the adenylate cyclase this will lead to the production of the cyclic AMP, and this will activate other proteins like the protein kinase A, and this activation will lead to the phosphorylation of the flagellar proteins, and as a consequence to a hyperactivated movement of the sperm cell. And from another side, this will lead to the removal of the cholesterol will lead to the membrane fluidity and the unmasking of the sperm cells, and also the increase in the calcium levels, as with the entry of the calcium will induce the unmasking of the receptors. So we have two elements that will lead to the unmasking of the sperm receptors, which are the redistribution of the phospholipids, yani the membrane fluidity, and also the increase in the calcium level will lead to the unmasking of the sperm by modifying the oligosaccharide chains of the surface proteins. Okay. Now what
دكتور
are
بس إذا فكرة إنه السبرم بايندينج specific to species أو لا ما فهمت.
يعني sperm cell of the human, for example, why it cannot recognize or bind, recognize another oocyte from another species. لأنه to recognize this oocyte, أول شيء the sperm cell should bind to its zona pellucida, okay? But the receptors, مش قلنا عنا receptors على ال sperm cell that are specific to the glycoproteins in the zona pellucida.
Yes.
The receptors of the sperm cell cannot recognize the glycoproteins of the zona pellucida of another species. يعني بس هن رح يتعرفوا على تعول humans. يعني whole receptors are specific for the glycoproteins present in the zona pellucida of the same species. Okay. If it is another species, the receptors on the surface of the sperm cell cannot recognize the glycoproteins بالزونة pellucida تعول whole ال other species.
Okay, doctor. Yes, clear. Thank you.
Okay. So what are the capacitation outcomes, يعني, or the results of this capacitation? So the membrane modifications allow the occurrence of the following events. We will have a hypermotility of the sperm cell, as we said, the preparation of the acrosomal reaction, okay, as a result of the capacitation. We said that the cytoplasmic membrane is weakened, and this will prepare the cell for the acrosomal reaction, and the unmasking of the protein of the receptors, of the sperm receptors, so the release of membrane protein sites for recognition of the zona pellucida and the oocyte membrane. Okay, so the unmasking, we want to say here the unmasking of the sperm receptors that will allow the recognition of the zona pellucida and the oocyte membrane. Now, we had the capacitation. Now, this capacitated sperm should reach the ovulated egg سوري انقطعت الكهرباء عندي. I will continue. Now this capacitated sperm, now we said in the uterus and in the fallopian tubes, we will have the capacitation. This capacitated sperm, this sperm is now capacitated. So if it will reach the oocyte, it can recognize it, and it will be able to interact, to recognize it and to interact with it and to fuse with it. Okay, if the capacitation didn't occur, even if the sperm cell will reach this oocyte, it will not be able to interact and to fuse with this oocyte. We wouldn't have a fertilization. Now, this capacitated sperm reached the ovulated egg, and after crossing the uterus and the tube, where it was capacitated, it will reach the oocyte here in the ampulla, where will be the fertilization, where the fertilization will occur. Now, the sperm cell this is the oocyte released in the ampulla. As we said, we have the oocyte surrounded by the zona pellucida and the corona radiata cells and the cumulus oophoris.
دكتور
Okay. So those sperm cells they will first find the cells of the granulosa, the cumulus oophoris, and the corona cells. They should first of all cross those, remove those granulosa cells, and then
دكتور صوتك عم يقطش.
yes.
ايه دكتور كتير يعني مش واضح الحكي.
It's from the internet connection. إذا ضل يقطش maybe I will record also the rest of the session, okay? لأنه I can't do anything for the connection.
دكتور حتى انت وعم تحكي هلا كان عم بقطش فما سمعنا شو عم بتقولي.
طيب هلا بعده عم بقطش.
يس دكتور.
راح الصوت.
هي عملت ميوت بركي راح تضبط الانترنت.
يس.
راح الصوت شي؟
ايه
Okay.
لا دكتور بعده عم يقطش.
يا دكتور صوتك بعده ما بين.
دكتور ما بعرف إذا عم تحكي بس نحن مش سامعينك أبدا.